So, introduction to trait-based ecology. So what I would like to do today is introduce why a trait-based approach in ecology. Why do we want to have a focus on trait-based ecology? I'll try to give you a short little history of trait-based ecology. We're going to start probably what's going to be a long discussion of what is a trait and how do you measure traits. And I just want to introduce a little bit of this very important question: what causes traits? Okay. And if you haven't done so already, please make sure to mute yourselves. That would be great. All right, so why trait-based ecology? All right, so if we kind of step back from the world and we step back and we think about the biodiversity on the planet, given the series of environmental crises that the planet then is facing, as biodiversity scientists, we actually have a responsibility to try to predict to the best of our ability how the biosphere is going to change and be modified to future climate changes as well as land use changes. And in general, this kind of demands on us as ecologists and biodiversity scientists to develop more of a predictive biodiversity science. And so that's where trait-based ecology comes in, to try to kind of edge us closer to a more predictive biodiversity science. Okay. All right, so let's step back, big picture. Let's go to the dictionary. And in particular, if we look at what a trait is, a trait can be formally defined in terms of what a phenotype is. Okay, so we have to understand what a phenotype is. And so the definition of a phenotype is an observable property of an organism that's produced by the interaction between the genotype and the environment, right? So when we study traits, we're really studying the phenotype. And the phenotype then is a constellation then of traits, right? So then the phenotype is going to include all these different attributes of an organism that's going to influence then how that organism survives, reproduces, and interacts with the environment, right? And so traits then are going to enable us to make these linkages between organismal survivorship, reproduction, and the interaction then with the environment. And so in general, in ecology and evolutionary biology, the phenotype is kind of like our fundamental unit. It's what we focus on and what we study, all right? And so we're really interested in the diversity of phenotypes, and traits are how we then describe phenotypes. All right, so why traits? So I'm pulling a quote here from a paper by Paul Ketty in 1992. And in this quote, Paul says, "Statements about traits give generality and predictability, whereas measurements of species richness or species diversity, in contrast, tends toward contingent rules and special cases." Okay. So this kind of sets the stage for why we measure traits, because we're interested in generalities. We're interested in ultimately in prediction. All right, so why traits? Many years back in 2006, I worked with this lead author, Brian McGill. He was a graduate student at the time here at the University of Arizona. And we worked with a few other co-authors here. And we tried to put together what we felt was kind of an emerging series of questions in ecology. So back in the early 2000s, there were a series of questions that were kind of revolving around kind of trait-based ecology. And so we were kind of struggling to make sense of what trait-based ecology was. And so this paper was our attempt to do that. And in that paper, if I want to just pull out kind of a quote or two. And so in this paper, we say that although being interested in the role of traits in ecology is not new, and trait-based ecology isn't new. You can go back to Humboldt and Darwin, and people have been measuring traits for a very long time. But what seems to be new now is that, in general, there is a new need for traits. And the need for traits has to do with this urgency to generate generalities and predictions. And so kind of historically, ecologists have kind of preferred to emphasize this nomenclatural approach. That is an approach of going out in the world and putting names then on species and looking at species diversity. Okay? So this nomenclatural approach then focuses on species identity. And in general, we think that this has resulted in a loss of ecological generality. That is, it's not a very strong measurement about the world. But in contrast, if we focus on traits, statements about traits give more generality and predictability, whereas this nomenclatural approach to ecology tends toward being highly contingent. That is, so we talk about contingent rules and special cases. Right? So this is kind of the background. All right, so why trait-based ecology? So traditional measures based on species richness don't really adequately capture the predictions then that are needed within our models, or then what we want to predict about the future. And traits then enable us to directly link how species perform in different environments. And so we want to know how well different species will perform in different temperatures or in different precipitation regimes, or in the presence or absence then of fire. And so in general, traits then enable a more predictive ecology. So that is why we focus on trait-based ecology. But then in general, although we're not going to have a chance to talk too much about more kind of the theoretical aspects in this lecture, traits enable us to develop more quantitative and mechanistic theory, and we use that then to make predictions. All right, so I want to step back and kind of put trait-based ecology in a broader context. So within ecology, one kind of persistent question is, how does species diversity influence ecosystem functioning? And there's a very long line of papers and researchers who have researched and proposed then this question. But in particular, I wanted to point to kind of this classical debate in ecology between people like David Tillman and also Philip Grime. And so the idea here is that biodiversity is important because it influences how ecosystems function. Okay? Now, this debate has focused on how one actually makes this connection between biodiversity and ecosystem function. And this paper by Tillman, so I'm going to focus in on this paper by Tillman, there's this very nice graphical model. And in this graphical model, what Tillman and all do is imagine species as these circles in niche space. So hopefully everybody can see my cursor here. And so each of these different circles then is a species in niche space. And this niche space here is given by differences in temperature, going from low temperature to high temperature, and differences in soil pH. Okay, so you can imagine then different species then kind of specializing in different temperature and soil pH, right? And so the assumption then of this work is that as you add more and more species in a community or in an ecosystem, that those species then are going to start occupying more and more of this overall niche space. And the assumption is that as you occupy more and more niche space, you can produce more and more biomass or utilize more and more resources. So based on this simple kind of theoretical model, then there should be a relationship between the number of species in a community and the total portion of niche space covered, but then also the total amount of plant biomass then present, or the amount of biomass then that's produced, or the net primary productivity. So that there should be an immediate linkage here between species richness, the total niche space, but then also the total functioning then of an ecosystem. And so then that's the argument, that there's this linkage between richness and then niche space. Now, part of the issue here is that if you measure species richness alone, it's very difficult to say something about these niche axes, that is the temperature affinity then for a species, or a soil pH affinity for a species. That is the direct linkage between richness and niche space, or species performance in niche space, is very difficult. We have to make an assumption. And so another kind of issue with this biodiversity ecosystem functioning work is that if I pull out just a relatively recent paper here, this is a kind of a consensus of knowledge of the relationship between biodiversity and ecosystem functioning, is that if we look at all of these different experimental studies, and this paper kind of presents this plot of the total number of experimental studies at the time, looking at the relationship between number of species and a measure of ecosystem function, that is the total amount of plant biomass present, what they find is that in general, yes, they tend to find these positive correlations, but I think you can all agree that the strength of that correlation is really low. That is, there's a lot of variation across here. So the relationship between species diversity and ecosystem functioning, while there looks to be a signal, it's not a very strong signal, and it's not a very impressive signal. And this isn't enough to basically make a big deal out of, right? It doesn't look like it's a very strong signal. And so the expectation here is that we can do better in making the linkage between diversity and ecosystem functioning. And we can do better by, instead of looking at species richness, instead look at measurements of traits themselves. Okay. So I'm going to skip this part. All right. So if we step back from trait-based ecology, there's this overall thesis, or this underlying kind of assumption, big assumption, that progress in biodiversity science has been limited by the primary focus on species richness, that is the number of species per unit area. And instead, we want to try to look at other patterns of species diversity that have a stronger basis, to make a stronger link between diversity and ecosystem functioning. Okay, so alternative measures of diversity. So I wanted to point you to this very nice chapter. It's in a book called Frontiers of Biogeography, New Approaches to the Geography of Nature. It's actually from paleontologists here, but the title then of the chapter is Beyond Species Richness. And in this chapter, they do a very good job in arguing that a true understanding of the processes underlying diversity patterns requires a better understanding of other aspects of organismal biology and geographic variation in these characters. And the point that is emphasized here is that if you incorporate additional information about species diversity, such as the diversity of morphologies, the diversity of functions, and even phylogenetic affinities of species, that's more of a true and kind of more of a reflective way of looking at the diversity of life. So in other words, instead of just looking at the number of species present as a measure of species of diversity, we need to look at the diversity of morphologies, functions, and also phylogenies as well. And so for this course, we're going to be looking at one important aspect of these additional measures of richness, the diversity of traits, which is linked to the diversity of functions and the diversity of morphologies, but is also linked to phylogeny, but a few steps removed. Okay. So what's our central hypothesis? Okay, and so one kind of central hypothesis in trait-based ecology is that the patterns that we see in the changes in the abundances of traits and the diversity of traits, but also in the dispersion of traits, that is, whether or not traits tend to be really abundant or really rare, if we measure these additional metrics of trait diversity, these can better reveal the processes then that are ultimately structuring biological diversity and how that diversity will respond in the future to any sort of environmental change. All right, so why measure functional traits? Well, we can develop mechanistic linkages, but we can also link better functional diversity to ecosystem processes. But also, more importantly, and I'll emphasize this in a little bit later, is that we can make quantitative comparisons then between different places in the landscape, different points of time, and so on. And so one thing that we'll talk about in the course is that the data that we can collect, we can use then these data to compare to other elevational gradients and to other data sets available in the literature, but also that we can collect and in the future, right? So we can then compare our different locations. All right. So just went over why trait-based ecology, a brief overview, but now let's talk a little bit more about the history of trait-based ecology. All right. So as in any kind of field of subdiscipline in science, we often have a series of different assumptions. And I want to highlight what I think is one of the prominent assumptions in trait-based ecology. And that assumption is diagrammed right here. And actually, we can kind of trace this to several different kind of authors. And so I took this figure from this paper by Viola et al. 2007, who I believe took it from Kingsall and Huey in 2003, who took then their figure from Arnold in 1983. And probably Arnold took this figure from somewhere, but I haven't figured that one out yet. So this is a complicated figure. So let me try to walk through this. And so what we have here at the top are a series of different measurements of traits. And so our traits are different values of Z. So trait 1 or Z1, Z2, Z3, many different traits that we can measure. But all these arrows and linkages are describing an organism, okay? So those traits then comprise the phenotype of the organism, and those traits then are going to interact with each other, so they could be correlated with each other. But then those traits then are going to ultimately influence a performance measure of the organism, okay? So those traits are going to influence how well an organism performs in a given environment. Okay, so what do I mean by a performance measure? So this could be the growth rate of an individual. It could, if you're talking about animals, it could be how fast maybe a lizard can run. So you can measure sprint speed, for example. It could be a measure of metabolic performance or your metabolic rate, for example. Okay, so traits then influence the performance of an organism, but then ultimately the performance of an organism is going to be influencing its fitness. Okay, and so remember, fitness then is going to be comprised of both survivorship and reproduction. All right, so how well an organism survives in a given environment, how well an organism reproduces in a given environment. Okay, so there's this linkage, this hierarchical linkage going from traits to performance to fitness. Now, also within ecology, these linkages are within an individual and then ultimately a population. But then in ecology, we want to make the linkage between traits and biodiversity, ecology, and ecosystems. Okay, and how we do that, how we make this additional linkage, that's actually up for debate, and we're struggling to do that right now. So a lot of questions in trait-based ecology is how do we make this last linkage? Okay. All right, so let me highlight a few kind of research focal areas. And I've tried to simplify this as much as possible, and with the eye of trying to point you to some key literature, some key papers that, that if you're interested, you can read up on. And I'm definitely missing over and I'm glossing over a lot of different papers here. So if I don't include your favorite one, my apologies. Okay, so kind of the first area that I want to highlight is research associated with trait dispersion. Okay, and so dispersion is a word that indicates how, if you array traits along an axis going from low trait values to high trait values, how clustered or dispersed then those traits are along a trait axis. Okay, so we'll give a few examples of this. All right, trait dispersion. All right, so I wanted to kind of point out that a lot of these questions actually kind of go quite a ways back, and so I wanted to pull out some quotes from Charles Darwin here. So in his book on the origin of species. And so what Chuck has this great quote here. And so he says, "The truth of the principle that the greatest amount of life can be supported by great diversification of structure is seen under many natural circumstances. In an extremely small area, especially if freely open to immigration, where the contest between individual and individual must be severe, we always find a great diversity in its inhabitants." Okay, so in this quote, what he's talking about is he's focused in a small ecological community, and he's looking at the diversity of species within this community. And actually, if I remember right, this is a plant community. And so if you go to the beginning of the quote here, and he says that if we look at this diversity within this community, he says the greatest amount of life can be supported by the great diversification of structure. That is, if you observe a highly diverse community, that is a community with a lot of different species, that's reflecting the fact that you have a great diversification of structure. Structure in what he's referring to is differences in morphology, differences in traits, basically, differences in phenotypes. And so he links this diversification of traits or structure with this contest between individual and individual. So he's immediately making a linkage between what he means by contest here. According then to Darwin, this is competition. All right, so as competition increases, that then is the fuel or the mechanism by which we get this diversification of structure and increases in diversity. So he's making this linkage between competition the diversity of traits, and then the diversity of species. Okay. And so he gives this one example. And so he was looking, I believe this is for a meadow in England, and says, for instance, I found that in a piece of turf, 3 feet by 4 in size, which had been exposed for many years to the exactly same condition, supported 20 species of plants, 3 belonged to 18 genera and to 8 orders, which shows how much these plants differentiated from each other. So when he's talking about differentiated from each other in terms of diversity, it's not differences in genera and orders and species, but this differences he's referring to is the differences in structure, morphology, and traits. And so Charles Darwin makes this direct linkage between diversity and traits. Okay? So these ideas go quite a ways back. Now, if you're interested in this linkage between traits and diversity and species diversity, this is where niche conservatism comes in. I'm not going to talk about niche conservatism, but for those of you interested, this is where you may be finding some interesting linkages. Okay, so within community ecology, when we look at the diversity or dispersion of traits, we can talk about the dispersion of traits in terms of how the number of individuals belonging to different species. And so here, the different species are species A, B, and C. We can array them, these different species, along trait axes. And so this is a given trait value. Maybe this is leaf size or seed size going from low to high. And so here we have 3 different species. And in this case, species A, B, and C are dispersed kind of evenly along the trait axis. And so we tend to call this the traits are being over-dispersed along the axes. That is, species are more evenly distributed along this trait axis. This type of pattern has been linked with abiotic interactions, in particular competitive interactions, but also more recently, we can link this to patterns in predation and even possibly either mutualistic patterns or relationships with herbivores or other pathogens. Okay, so this would be a trait over dispersion. Now, it could be entirely possible that traits then of individual species don't necessarily, are not involved in terms of how species interact with each other. And instead, maybe traits are kind of randomly dispersed. That is, there is no specific pattern to how traits then are dispersed along a niche axis. And so we call this a random dispersion of traits, which could be consistent with a neutral expectation. That traits really are not important in terms of how species then are packing in or related then to each other. Another pattern of trait dispersion is something known as abiotic filtering. Abiotic filtering is an interesting word. And so the abiotic environment, remember, deals with the environment where species or individuals then interact with. And so this could be temperature, precipitation, soil pH, or something like that. And so if the local environment then selects for a limited subsets of traits then that do very well in the environment, then we should get a clustering then of traits in trait space. And so abiotic filtering then should be characterized, according then to this literature, to more of a clustered pattern in trait dispersion, okay? So these are 3 examples of trait dispersion: trait over dispersion, random dispersion, and clustering, okay? And so in trait-based ecology, for community ecology, we tend to look at whether or not trait patterns are over dispersed, random, or clustered. Okay. So if you want a very nice paper that kind of provides you an overview with talking about different patterns of trait dispersion, I'd recommend this paper by Philip Grime, published back in 2006. And he talks about different ecological processes that will lead to trait convergence or trait divergence. Okay. And so again, in community ecology, and sometimes also in population biology, trait-based approach, we're often talking about convergences of traits or divergences of traits, and what that may mean in terms of the underlying processes. Okay. All right. So I also wanted to point you to this paper, which I believe some subgroups may be reading. All right. And so this process of measuring trait dispersion then could actually reflect multiple different processes. Okay. And so this diagram is pulled right out of this paper. And so we can think of ecological processes as operating on several different ecological and evolutionary scales. And so we can talk about the regional pool of species. And so this is more at the biogeographic or regional or ecosystem level, where we have several different species then that are present within a given biogeographic realm or within a given ecosystem. And if we look at the trait values, the mean trait values of those species in the regional pool, you can see that they're dispersed across that trait axis. Now, as those species then disperse within a local community, if there is any sort of process that limits those traits then that can get into a given community, that will then limit the amount of variability of traits, that is filter then those traits that are found locally. But then as those species, once they arrive by immigration, as they start interacting then with each other, a certain trait value may become selected against or become very rare, so that the dispersion then will change, right? So we go from a clustering pattern, and then once we get this clustering then of traits, we could then see an over dispersion, but of those limited kind of clustered set of traits, okay? So we can talk about clustering and over dispersion at several different ecological and evolutionary scales, ranging from the regional pool down to the local community. All right, so functional diversity. All right. Another aspect of kind of focus on traits is associated with functional diversity. And so this is a really broad literature, and I only want to give just a few kind of quick examples. So one way of studying functional diversity is to talk about trait hypervolumes or functional hypervolumes. And so here I can kind of describe a functional trait hypervolume by, if we plot, so each data point here is a different species, but each species is then going to be characterized by, here in this case, 1, 2, 3 different traits. All right, and so these traits then of a species is this is the density of wood, the mass of the seed, how big the seed is, and here a measurement on leaves is the specific leaf area. That's the leaf area per unit mass. And so here are all the different species then found then within the community. And if you notice, all of those species are occupying this trait volume. So if we put, in this case, a convex hull, or if we kind of wrap, put plastic and kind of wrap all of those species together, we get this volume, right? And so this is one measurement of trait diversity. And so we call this a trait hypervolume. That is, how big of, in phenotypic space, then do these species occupy? And so what's nice about this is that we can compare different communities or different populations, or we can even compare species and look, and so the volume of trait here would be the amount of intraspecific variation. But at the community level, we're looking at interspecific variation. Okay, so that's a quick introduction to the trait then hypervolume. Okay, at very large kind of biogeographic scales and elevational gradients, ecological gradients, we can extend kind of biodiversity theory to focus on functional hypervolumes. And so there's a long line of literature that is focused on why we see differences in species diversity as you go from highly diverse communities to really species poor communities. One hypothesis for why some species have more, why some communities have more species than others is something known as the biotic pressure hypothesis. And the idea there is that in warm and wet environments, natural selection has led to an increased range of phenotypes. And so this could be reflecting differences in the amount of competitors, enemies, diseases. So in really warm, productive environments that would favor high rates of competition, the evolution of more disease vectors and more pathogens, and because of which that would then lead to a diversification then of species, which could then lead to more functional traits, a wide range of functional traits or bigger hypervolume. Another kind of opposing hypothesis is the stress dominance or filtering hypothesis, saying that in more stressful environments, and so in really species poor environments, you would then expect stronger filtering or stronger stabilizing selection, and that would place more of a limit on the functions or traits then present, okay? So immediately we can cast different hypotheses that have been present in the literature for a very long time, but based on species richness, we can recast those hypotheses in terms of traits, right? And so now there's quite a few studies trying to do this. Okay. So kind of the next area that I want to focus in terms of the background of what people use trait-based ecology to look at is something that I kind of characterize as the diversity of life histories or ecological strategies. And if I were to point you to one paper that's kind of classical plant ecology that focuses on this, this is another paper by Philip Grime in 1977, something known as the CSR triangle. And so this, if you haven't had this before, this can seem really odd and complicated, but it's actually very simple. And what the goal here, according to Grime in the CSR triangle, is that differences between species can be characterized by different strategies. So when we step back and think about differences between species, those differences in species can be cast in terms of differences in strategies. That is, species are going to respond differently to levels of competition. So you can go from low levels of competition to high levels of competition. Or species can differ in terms of how they respond to disturbance. That is, they tend to like a lot of disturbance or very little disturbance. But also some species can deal with high levels of stress in the environment or low levels of stress. And so the idea from Grime and in his CSR triangle is that each species then is either a good competitor or a bad competitor, but then will respond differently to disturbance and stress. So each species is going to be kind of found in this space of competition, disturbance, and stress, right? And so it's a way of characterizing the strategies then of species. Okay? And so kind of through the years since this Grime paper, 1977, people have been trying to characterize where different species then tend to fall into this CSR then triangle, right? So trees tend to be really good competitors, right? And don't like a lot of disturbance, whereas annual herbs and things tend to like a lot of disturbance, but deal with very little stress, and they tend not to be really good competitors. Okay. So that's very nice in terms of how we start to classify, but can we do better at characterizing the diversity of life histories and strategies? And so this is where a trait-based approach has come in. So kind of over the years since the work of Grime, a lot of researchers have tried to redo this work, but in terms of traits. Okay? And so I would point you to these two more recent papers that are basically redoing the original Grime work, but in a much more kind of quantitative and trait-based way. And so what these papers kind of emphasize is that there is a continuum of life history variation across species. That is, all different species then on the planet can be characterized by species with slow life histories, that is characterized by slow traits, or species with fast life histories, and those species are characterized with fast traits. So the idea is that the traits are telling us where species are located in terms of their different strategies. Okay? All right, so here is kind of one set, an attempt here to kind of put then species along this fast-slow continuum. All right, so if you're going to be working in Sean Michelitz's group, he's likely going to talk a little bit more detail about this. And so we can take a series of different leaf traits, such as photosynthetic rate, respiration rate, nitrogen, phosphorus content, how long a leaf lives, measurements like the specific leaf area, but also these more thermal performance measurements, and relate them to this fast-slow continuum, okay? And so again, the idea is that with the different species we're going to be encountering, we can characterize their physiology, their nutrient content, some aspects of their life history, and even how they respond to temperature along this fast-slow continuum. Okay? So to give you an idea of how this fast-slow continuum kind of fits in, I'm giving you one example from Peter Reich's 2014 paper. And so what Peter did in his paper was pull together several different data sets. And in this data set, each data point is a different species. And if you look across all these different species, there is this cross-species inverse relationship between the survival rate and the growth rate. That is, some species actually grow really slowly. So this group of species down here are growing really slowly, but if you notice, they tend to have high survivorship. That is, they tend to live a long time, but they die really slowly. But there's this other group of species down here that tend to grow really fast. We call these fast species, but they have a really low survivorship. That is, they're dying at a very high rate, okay? So we have this inverse relationship between life histories, okay? So survivorship, how quickly you're dying in the environment, versus how quickly you're growing in the environment. And so this is a kind of classic life history trade-off. And so we can characterize these different life history strategies, fast strategies and slow strategies, by measuring traits. So the traits then enable us then to identify the different types of strategies then that we find within a population, within a community, and within an ecosystem, okay? So here's to pull out a quote. So traits help explain differences in growth and survival across resource gradients, and thus the assembly of communities across light, water, and nutrient gradients. So different environmental factors will select for either slow traits or fast traits or something in between. And so according to Peter, the importance then of these fast and slow traits is that these traits are important because these traits will then scale up. Fast traits are associated with faster rates of ecosystem processes such as decomposition and primary productivity, and slow traits are associated with slow process rates. So then we can start to make some linkages with ecosystems. Okay. All right, so then to kind of finish up, I'm gonna skip over this part in the interest of time, but I will also make available this PowerPoint so you can look into this a little bit more. These additional papers also focus on this fast-slow continuum, this fast traits and slow traits, but they do a little bit more detail and looking at additional trade-offs than within species, all right? And so I would encourage you to look over this kind of in your own time. So I'm gonna skip over this part, but I'll make available this presentation as a separate file so you can look over that in your own time. So I'm gonna skip over this part. And so I wanted then to kind of bring us up to date then on kind of where we are in the study of the linkage between traits and functional strategies. And so I would point everyone to this recent paper by Sandra Diaz et al. That was published just a few years back. And this is called The Global Spectrum of Plant Form and Function. And this paper, I think, is what I like about it is this blob that's published, one of the main figures. And what this blob is reflecting is that what they did is they put each of these data points is one species, and they then measured all of these different traits kind of associated with these different symbols then that are arrayed around then the blob. So we have differences in leaf size, we have differences in nitrogen availability, differences in leaf area, differences in plant height or size, differences in wood density, tissue density, and many other different traits. And so what this blob is reflecting is the multivariate distribution. It's kind of a measure of hypervolume. So kind of thinking of this as a hypervolume in terms of how all these different species are arrayed in trait space. And what you immediately see is that there are two major axes of how species are differentiating then from each other in trait space. The first major differentiation between species is just body size. That is, whether or not you tend to be a very small plant or if you tend to be a very big plant. So effectively, size is our first important trait that we want to measure, because then that's going to be reflecting this primary difference in the fast-slow continuum is reflected then in differences in body size. But then the variation in this direction, up and down, in vertical direction, that variation reflects differences in what we call leaf economic traits, differences in nutrient content, differences in leaf size, differences in allocation or carbon allocation, differences in photosynthetic rates. Okay? So between body size and what we call these leaf economic traits, which we'll be getting into more likely in some of the subgroups, but also when we're in Peru, these are the traits then that allow us to differentiate differences in the vertical direction. Okay. All right. So the last kind of like major area of focus, and I'm only going to spend kind of one slide on this, is how to scale up from traits all the way to ecosystems. And we'll likely have more time to discuss this, but likely more down in Peru. And for this, I would also point you to another paper by Philip Grime, because he's a very good writer and has a way of kind of characterizing the overview of the literature. But this gets to ultimately this biodiversity ecosystem functioning debate. And Grime's way of making the linkage between biodiversity and ecosystem function is not to focus on species diversity per se, but instead to focus on the frequency distribution of traits within a different community. And according to Grime, the best way to link traits to ecosystem functioning is that we need to know the abundances of these different traits. That is, we need to know what trait is the most frequent in a community, and also which trait is the most rare in a community. So here is an example within a given community of a given trait, plant height, looking at the frequency distribution of individuals. Now, this isn't species, so this is individuals, of a given height value. And as you can see, there's a given height value or size measure that's the most frequent in a community. And we could do that for several different traits. And so according to Grime, what you want to identify for linking traits to ecosystems is the most frequent trait value. That is, we want to know the height that is the most dominant, or a measurement of the seed mass that's the most dominant, if we want to make then these linkages with ecosystem functioning. Now, there's theoretical reasons why we want to do that, but we can save that for another cut. Okay, so I want to try to end out here, and I'll try to scoot through this in my limited amount of time. I want to talk briefly about what is a trait. Now, within each of your different groups, this is likely going to come up, and depending on the different groups, we're going to then be focusing on different traits. And so this discussion will likely continue quite a bit, but I want to give you a quick introduction to what a trait is. And so let's make this try to make it simple. All right, so a trait is basically anything that you can measure in the phenotype, right? So we can probably think of a huge list of possible things that we can measure in a phenotype, right? Now, we often come up with this additional term of functional trait, all right? So let's start by thinking, what's the difference between a trait and a functional trait? Now, a functional trait can be very confusing to people because it has this term functional, all right? So what do we mean by functional? Well, a functional trait is a trait then that influences a plant function, all right? So remember, we mentioned performances, performance measure, or fitness, all right? So a functional trait is a trait that influences the performance of an organism or its fitness. And so you can think of a functional trait as a trait then that's linked to demography, right, to growth rate, or even to fitness, okay? So differences between traits and functional traits. All right, so if we were to fill in this diagram of traits, okay, to these different measures of how well a plant does, these different performance measures, and different aspects of fitness via demography, survivorship, and reproduction, right? A functional trait then is a trait that has a strong effect on performance and demography. That's what a functional trait is. All right. All right. So how do we measure plant functional traits? Within each of our different groups, we're going to be talking more about the methodologies that we're going to be using. All right. But if you haven't done so already, I would point you to this handbook and protocol for measurements of plant functional traits worldwide. I'm here showing you an older version of this. This is kind of one of the original ones that was published in 2003. There's now an updated version that goes into a little bit more detail. And so that was published in 2013. So here is the new handbook, okay? But it's still basically the same handbook, but it's updated. And so I would encourage you to look through this paper, because this is a literal laundry list of various traits that people have measured, all right? So you can kind of skim through here, all right? And we're gonna be pulling out a few of these traits that we're gonna be focusing in on the course, all right? And so this handbook actually gets pretty Byzantine at times, but if you're really into these different traits, definitely check it out. And so I'm pulling out one very complicated diagram from this paper. And so if you notice here, this is a list of many different plant traits. And in this table, what it says is this is how these different traits are then linked to then many different other kind of measures or questions that you may be interested in. That is how, for example, plants may respond to climate, or respond to CO2, or respond to soil, or respond to disturbance, or your competitive strength, right? Or your plant defense, and so on. So if you notice, there are all these different traits, okay, that have been shown to make these linkages, that is, are reflecting these different ecological or evolutionary processes, okay? So depending on your types of questions, some traits may be more linked than to others, okay? So here's a listing of these key traits that are often measured in trait-based ecology. So here's a series of different whole plant traits, such as size or tissue density, things like the leaf mass fraction, the root mass fraction. Here are several different leaf traits. We're gonna be going over these more in the course and within your subgroups, and so I don't wanna kind of belabor kind of any of these. There's also root traits. We may talk a little bit more about root traits. Let's kind of move on. We're also likely going to be talking a little bit more in the course about tissue isotopes, in particular carbon isotopes, nitrogen isotopes, and also oxygen isotopes. These are very good traits because they're reflecting different physiological processes within plants, and also help us characterize differences in ecological and evolutionary strategies. And so with a lot of the leaves then that we collect in our plant functional trait courses, we also do isotopic analyses of these later on. And so we'll have a chance to talk about what tissue isotopes also mean and how that relates to trait-based ecology. Right. So which trait should you measure, right? And so I'm gonna pull out a great quote here from the new handbook. It says, "No methods handbook can answer the question of what are the best traits to measure, because this strongly depends on the questions at hand, the ecological characteristics of the scale of the study area, and on practical circumstances." Okay? And so often, you know, when students start out wanting to do trait-based measures, like, well, what trait should I measure? It's like, well, it totally depends. And it depends on many things. And so what you want to do is step back and think about traits then that link to the performance then of the organism in the environment, right? And so depending on your different performance metrics, you want to think about which traits then are best linked to that, okay? So an easy way to do that is let theory tell you which traits to measure. Now, this is a whole separate lecture in terms of which specific theories then one should then go to, but this, again, is question kind of dependent. So there's many different theories that link how traits in the abiotic environment influence organismal performance, okay? And so if you have more of a background in ecophysiology, in carbon and nutrient economics, that line of thinking kind of tells you what specific traits then are very important for, say, the water potential of a plant, or the carbon balance or nutrient budget of a plant. Energy budgets, in particular temperature, is pretty critical. In Sean Michelitz's group, he's going to talk quite a bit about energy budgets in terms of how that relates to the traits you're going to be measuring in the field. Carbon and nutrient kind of content, okay, that's going to relate to kind of theories of optimization of growth, nutrient limitation, R-star theory, and so on. Allometry, biomechanics, hydraulics, demography, something known as trait driver theory, all of these different lines of literature that tell you which traits you should be measuring, because the theoretical basis tells you which traits then to measure. Okay, but we'll have more to say about that later. And so what I want to end with here is just an overview of what causes variation in traits. And I'm not actually going to be able to answer entirely this question, but I'm hoping that this is going to be a nice springboard or a baseline for you to launch into discussions within each of your different groups, but also to ask questions later once we're in Peru. All right, so what causes variation in traits? Well, we can measure variation in traits at several different levels, okay? And we're going to be doing that in the course, all right? So kind of traditionally, we have measured trait differences in terms of differences between species. And so originally in trait-based ecology, we have kind of focused on trying to characterize how species differ from each other in terms of their traits. And that has then led to studying interspecific variation of traits across really big kind of climatic gradients, okay, or differences in biomes, differences in continents, differences in elevational gradients, and so on. But increasingly, we're now also interested in understanding intraspecific trait variation. And so a lot of variation in interspecific variation is due to microclimatic gradients. So very local differences in soil temperature or soil moisture, or even biotic interactions that happen at a very small temporal and spatial scale. Okay. So a lot of different processes then are involved in influencing species level differences at large spatial, temporal, and evolutionary scales, but also we can talk about differences at small scales. So I wanted to highlight at least this, this paper that probably many of you have already read. And if you haven't read, I would definitely encourage you to read this literature. So this is a paper known as the Worldwide Leaf Economic Spectrum, published by Wright et al. In 2004. This is kind of an update to the original paper by Peter Reich et al. In 1997. So both of these papers kind of point to kind of a similar set of patterns associated with variation in leaf traits. And this is known as the leaf economic spectrum. And so this is related to this fast-slow continuum. And so this is a figure from the Reich et al. 1997 paper. And so what we're looking at here, we have several different graphs. And on each of these different graphs, we have a data point. And this data point represents a given species. But we're now looking at trait correlations between leaf photosynthesis, nitrogen, specific leaf area, and how long a leaf lives. All right, and so if you notice, of all of these different traits, so all leaves across the globe, and they did this by studying leaves in the tundra, in leaves in the desert, leaves in the rainforest. So we have species from all over the globe, and those differences in biomes are then related here by the different symbols kind of up above. And if you put all these different species from across the biomes of the world, and if we look at these traits, photosynthesis, leaf nitrogen, specific leaf area, the area of the leaf divided by the mass of the leaf, and the leaf lifespan, what we actually find is that all of those traits co-vary with each other. That is, if a leaf is characterized by a very short lifespan, it tends to have a corresponding specific leaf area, leaf nitrogen, and photosynthesis. So short-lived leaves tend to be really high in nitrogen and have high photosynthetic rates, whereas leaves that are very long-lived tend to have low amounts of leaf nitrogen and low rates of photosynthesis. Okay. So we see these differences in ecological strategies basically constraining how all of these different leaf traits then co-vary then with each other. So this is known as the leaf economic spectrum. And so we'll talk about more of the leaf economic spectrum probably within your subgroups and also down in Peru. All right, so SLA. So this is a trait that we'll likely be talking a lot about. And so SLA is the leaf area, that is the area of the leaf, then divided then by the mass of the leaf, that is how heavy then the leaf is. And so a low SLA leaf, think of it as a very thick leaf or a leathery leaf, a leaf that's very hard to break in a way. So it's a thin and flimsy leaf, is high SLA leaf. And so we have less mass, okay, per unit area, but it also has a high photosynthetic rate and a short lifespan, whereas a low SLA leaf has more mass per unit area, but it also has a low photosynthetic rate and a longer lifespan. Okay, So why then do we see these differences, right? Why do we see a set of traits kind of all associated with these fast traits and then slow traits? All right. So this is known as leaf economics, okay? And so I want to end with just a little brief introduction to leaf economics, okay? And this is the only equation I'm going to show in this, in this lecture today. And so the idea is that in different environments, as we go from deserts to alpine tundra to tropical forests, that natural selection in each of these different environments has shaped leaves to have a positive net return on investment. That is, if you're going to be a leaf that occurs in a given environment, that leaf then has to have a positive return on investment. Another way of thinking about that is that a plant is going to be investing in a leaf, and that investment is costing in terms of carbon and nitrogen. So it's going to cost a given amount of energy, a given amount of carbon and nitrogen to build a leaf in a given environment. And over the lifespan of that leaf has to pay back the cost it took to build it, okay? And so we can understand leaf economics by this very simple relationship, okay? And so if we think of the total amount of carbon gain of a given leaf in its lifetime, the total carbon gain of a leaf can be approximated as being equal to this equation. That is, it's the photosynthetic rate of a leaf times how long it lives, right? So remember, the photosynthetic rate is the rate at which carbon is being assimilated, times the total length of time the plant is photosynthesizing, so how long it lives. But then to make this dimensionally correct, this has to be divided by how much mass per unit area that leaf is. Basically, how much carbon is being invested into the leaf itself, okay? And so we can relate that to this quantity here, where the total amount of carbon gained over its lifespan divided by the leaf mass per unit area has to be greater than or equal to this amount of carbon then that was invested initially, okay? So the return on investment has to be greater than or equal to a starting amount, okay? So we call this the minimum lifetime leaf carbon gain. That is the amount of carbon then that has to be obtained by the leaf in order to break even, okay? And so if you notice here, what this says is that there are many different ways that natural selection can operate on leaf traits in order to increase this quantity on the left-hand side of the equation, right? So if natural selection is going to operate on a leaf in a given environment to make sure that it's greater than or equal to this minimum lifetime carbon gain, it can change the photosynthetic rate, it can change how long a leaf can live in an environment, but it can also change how much leaf mass per unit area then is present. And that sets up a very strong constraint in terms of how all of these different traits kind of co-vary with each other. So selection in different environments is going to maximize or minimize any of these different trait values as long as the total amount of carbon gain is greater than or equal to this minimum lifetime leaf carbon gain, okay? And so what's interesting is that it turns out that across all these different leaves across the globe, the value of this minimum carbon gain is very similar. And it turns out that for every 4 grams of carbon then that's assimilated per 1 gram of carbon invested in leaf, that this is an approximately constant amount of all leaves. That is, as we go from very short-lived annuals to tropical tree leaves, that trait correlations that we see between photosynthetic rate, leaf nitrogen, leaf lifespan, leaf mass per unit area, that's all kind of reflecting this ultimate constraint of trying to maintain a positive carbon balance in a given environment. And so we call this leaf economics, okay? So in different environments then, natural selection is trying to maximize fitness but via the economics, the carbon and nutrient economics then, I believe. Okay. So that was just a very kind of quick introduction. And so with that, I'm going to kind of skip on through to my conclusions here. And so I gave you an overview of why we're doing trait-based ecology, a quick overview of the history of trait-based ecology, started the discussion of what is a trait and how we're going to be figuring out why we want to measure different traits. But also, I hope I've kind of introduced you to some of the kind of underlying kind of discussions and our understanding about what may be causing variation then in traits to begin with. Okay. And so with that, why should we measure traits? Well, there's a lot of different reasons. And so it's a really exciting time in trait-based ecology. And in the process of this course, we hope to kind of express to you the multiple levels of enthusiasm for trait-based ecology. But in general, plant functional traits are going to give us better insights into how we understand how organisms interact with their environment, okay? And it gives us a deeper kind of insight in terms of the underlying processes. And so they're also going to be providing us with an understanding about functional diversity, okay? Instead of just focusing on species richness and species diversity, we're now going to be able to start talking about functional diversity and how then we can take functional diversity and link that to ecosystem then processes, okay? And what that may mean for differences in, differences in the rates of climate change, land use change, conservation implications, and so on. And so what's also exciting that we hope to also introduce you to is that in our plant functional trait courses, we want to be able to compare different ecosystems. And so over the last many years, we've been doing this plant functional trait course in many different environments. And so we want to then kind of compare all these different ecosystems, these different courses that have very little overlap in terms of their species richness. In fact, no overlap in species, but they do overlap in terms of these traits then that we're measuring. That enables us to compare. Okay, and so with that, I wanted to say thank you